Hello and welcome back to the Biochemistry for Health Sciences channel. In today's video, we shall discuss cellular adaptation and death. As you know, the human body is made up of cells. So during an injury, a cell may suffer some damage. If the injury is mild, this damage is reversible and the cell can come back to its healthy state. However, if the injury is severe or chronic, keeps on happening, is a persistent injury, then the cell undergoes damage that is irreversible. That will lead to an irreversible injury, and that cell eventually will die, mostly either due to necrosis, which is a pathological phenomenon, or due to programmed cell death, such as apoptosis. Apoptosis could be either physiological or pathological. Let's look at this a little bit deeper. So, given any cell in the body, we should appreciate that this cell is constantly interacting with its environment in the body. The cell will try to maintain its intracellular environment, conditions such as concentration of chemicals, osmolarity, pH of the cell, and all of these will be maintained within a narrow range of physiological parameters. This is called a steady state or homeostasis. And the cell will adjust its structure and or function accordingly to changing demands of the body. So this is what a normal cell typically does. Now, what happens if the cell is stressed? By stress, we could mean physiological stress, for example, fear, lack of sleep, or by stress we could mean pathological stress or injury. And typical pathological stress includes hypoxia, ischemia, harmful chemicals or toxins, physical insults such as ultraviolet radiation or X-rays, infections by microbes, by bacteria, viruses, parasites, so on, abnormal immunological reactions, genetic defects, and nutritional imbalances are all examples of pathological stress. So a cell that is stressed will adapt to the stress. It will establish a new steady state. So let's look at this adaptation. What kind of adaptation do the cells undergo? One form of adaptation is hypertrophy. In this case, the cell size increases by increasing the amount of chemicals in the cell. As more and more cells become hypertrophic, the organ will also increase in size and undergo organ hypertrophy. Hypertrophy could be either physiological or it could be pathological. Physiological hypertrophy could be due to increased functional demand or what we call workload. For example, we see this in skeletal muscles that face an increased functional demand or workload, they undergo hypertrophy, and you see this, for example, in weightlifters. Another example of physiological hypertrophy is uterus hypertrophy that we see during pregnancy, and this happens because of stimulation by signals, by hormones. An example of pathological hypertrophy is hypertension. So in this case, the normal heart, when the blood pressure is high, this normal heart is constantly facing a pressure overload, and that results in increased size of its myocyte cells. And as more and more myocytes become bigger and bigger, the heart also increases in size, what we call cardiac hypertrophy. And of course, there is a limit. If this keeps on happening, eventually the heart will stop working, and what we have is heart failure. Another way the cell can adapt to the changing environment or to the stress is hyperplasia. So in this case, we are increasing the number of cells, not the size of cells, the number of cells, through cell division. Once again, hyperplasia could be either physiological or pathological. An example of physiological hyperplasia is seen in the menstrual cycle. The hormone estrogen will stimulate the uterus, so increasing estrogen concentrations will stimulate the uterus. The endometrial cells in the uterus will start dividing, so there will be hyperplasia, and that will lead to an increased thickness of the endometrium. And you can see the size of the endometrium increasing due to increased cell division or hyperplasia. An example of pathological hyperplasia is the condition called benign prostatic hyperplasia or BPH. In this case, there is excessive androgen stimulation of the prostate gland, and that leads to increased cell division or hyperplasia, and the prostate gland weight increases almost 3 to 5-fold. Another possible adaptation is atrophy. So in this case, there is a decrease in the cell size due to loss of cellular chemicals. An example of physiological atrophy is disuse. For example, during prolonged bed rest, the skeletal muscles can actually decrease in size, and that's called muscle atrophy. Example of pathological atrophy is when the nerve that is supplying information to the muscle is damaged. For example, the nerve is cut, and that leads to muscle atrophy. Another adaptation that a cell can go through when stressed is called metaplasia. Metaplasia is a reversible change where one cell type, for example, a columnar cell, is replaced by another cell type, for example, a squamous cell. So a columnar cell could be transformed into a squamous cell. And the reason this happens is because the newer cell type that is formed, in this case the squamous cell, will be better suited to tolerate the abnormal environment or the new environment that the cell is seeing. Once again, metaplasia could be either physiological or pathologic. An example of physiological metaplasia is what we see in the cervix during puberty or pregnancy. So during puberty or pregnancy, there is increased concentration of estrogens as well as the hormone progesterone. These hormones stimulate rapid cervical growth. And this causes the normal columnar cells that are found in the endocervix to be pushed outward into the ectocervix. So the cells here in the endocervix are now going to be pushed more towards the ectocervix part, the ectocervix area right here. So these columnar cells in the endocervix will be replaced by squamous epithelial cells. And this happens because the ectocervix is facing conditions in the vagina. And in the vagina, we have a low pH, an acidic pH. So these squamous cells are better suited to stand the low pH of the vaginal environment compared to the columnar cells. So this conversion of columnar cells in the endocervix, what we call the transformation zone, into squamous epithelial cells, is an example of physiological metaplasia. Now, physiological metaplasia is not a very common occurrence that we see. Pathological metaplasia is more common, and an example shown here is what happens in the lungs of a smoker. The normal respiratory epithelium is made up of columnar cells that have cilia. So these cells in the lungs of a smoker are replaced by squamous cells. They undergo metaplasia. So the columnar cells transform into squamous epithelial cells. The good news is that these squamous epithelial cells provide better protection against the irritant, against the smoke. The bad news is that these cells don't have the cilia, so the protective mucous barrier is lost. If metaplasia continues, it can lead to what we call dysplasia. In that case, there is loss of uniformity. They are very highly variable cell shapes and sizes, and there is an altered tissue architecture. We shall discuss more about this in a future video on cancer. So up to a certain point, when the stress is eliminated or the injury is mild, the cell can go back to its original steady state. However, if the limits are exceeded or if the cell is severely injured, then the injury is irreversible and the cell undergoes either necrosis or programmed cell death such as apoptosis. Let's now see what happens in a cell that is injured. We will use ischemia as an example because this is a very common cause of cell injury. Now we should be able to differentiate hypoxia from ischemia. Now in hypoxia, only the oxygen supply to the cell is compromised. However, in ischemia, the oxygen as well as the fuel, in this case the nutrient, supply to the cell will be compromised. Ischemia is mostly due to a mechanical blockage of the artery that is supplying blood to the tissue, and sometimes it can also happen due to reduced venous drainage, what we call venous congestion. So let's see the sequence of events that happens, let's say, in this cell. This cell is being supplied blood by this blood vessel here, and let's say there is a clot, a thrombus or clot, and this clot is completely blocking the blood vessel. So now there is no nutrient supply as well as no oxygen supply to this cell. So let's see what happens to this cell once this thrombus forms and stops blood supply to the cell. One of the first things that happens within 1 to 2 minutes of the blockage is the function of the cell is impaired. So if this cell was a myocardial cell, it would not be able to contract anymore. In about 2 to 3 hours, then the ultrastructural morphology of the cell will change. So we can only see these changes under an electron microscope. Now, 6 to 12 hours later, we can see changes in the cell using light microscopy. Now, recall from the videos on metabolism that the gas oxygen is very important in order to generate maximum amounts of ATP. So if oxygen concentration decreases due to the blockage, oxidative phosphorylation in the mitochondria is going to decrease, and that will lead to a decreased concentration of ATP in the cell. Now, even a 5% decrease in ATP generation can disrupt several cellular activities. Now, one of the proteins that use a lot of ATP in a cell is the sodium-potassium pumps. These pumps pump sodium out of the cell and bring potassium into the cell. Now, without sufficient energy, without sufficient ATP, the function of these proteins, these pumps are going to be affected. So these pumps will start slowing down. So the decreased activity of the sodium-potassium pumps due to insufficient ATP is going to cause sodium to now enter the cell. And when more and more sodium enters the cell, the sodium will increase the intracellular osmolarity. So the higher osmolarity inside the cell compared to the outside is going to make water enter from the outside towards the inside of the cell. And this water getting into the cell is going to make the cell swollen. So that leads to cell swelling, which is one of the early things we see when a cell is undergoing injury. So in a swollen tissue, we see a pale color, pallor, and that's due to the water that is compressing the capillaries. And that's why we don't see the red color anymore. We see a very pale color in swollen tissue. And with all the water present in the tissue, that's going to increase the pressure in the tissue, what we call increased turgor. And of course, with all the water present, the weight of the tissue is also going to increase. So without oxygen, most of the energy now will be generated through glycolysis. Okay, so basically the cell is going to break down glucose into pyruvate, and if it has enough glycogen, it will break down the glycogen into glucose and then the glucose into pyruvate, and that happens through glycolysis or glycogenolysis, breakdown of glycogen to glucose and then glucose to pyruvate. So now the cell is going to rely on the ATP that is generated in glycolysis for survival, because it has no more oxygen, so it cannot use the energy generation system in the mitochondria. Now recall from metabolism that for glycolysis we need NAD. We need NAD+. And since NAD comes from a vitamin niacin, it's in limited supply. So as we discuss in metabolism, in order to make sure that glycolysis can keep on happening, we have to convert the NADH that is formed in glycolysis, back into NAD, so that this NAD can be used for glycolysis. And this is done by the enzyme lactate dehydrogenase, or LDH, which takes the pyruvate that is formed in glycolysis, converts the pyruvate into lactate, and in the process NADH is converted to NAD, and the NAD can be used by glycolysis. Remember, as we mentioned and emphasized many times in previous videos, energy, energy, energy. So the cell is going somehow to make sure that it can generate enough energy. So by generating energy now through glycolysis, what it is producing is this product here, lactic acid. Now more and more lactic acid will accumulate in the cell, and lactic acid, although it's a weak acid, is going to accumulate and make the cytoplasm acidic. The pH is going to decrease. As the pH decreases, that's going to affect several enzymes as well as protein function in the cell. Also, DNA, note that DNA is negatively charged. As the pH decreases, these negative charges will decrease, and that will cause chromatin clumping, because earlier with normal pH the DNA is negatively charged. The negative charge repels the DNA from another DNA, but now as the negative charges are neutralized, the DNA start coming closer to one another, and that leads to what we call chromatin clumping, another thing we see in a cell early on during cell injury. Now recall that the calcium in the cytoplasm, the calcium concentration in the cytoplasm, which is about 0.1 micromolar, this is 10,000 times less than the calcium that you find either in the extracellular space or in the ER. So normally calcium concentration in the cytoplasm is held at very, very low concentrations. And this is done mostly by calcium pumps, calcium ATPase pumps. These pumps pump calcium out of the cell or into the endoplasmic reticulum. And without ATP, due to ATP depletion, these calcium ATP pumps will also start slowing down, and that is going to cause an increased cytoplasmic calcium. So the concentration of calcium in the cytoplasm is going to slowly start increasing because these pumps are not working anymore because there's not enough ATP. One of the effects of this increased intracellular calcium is on the mitochondria. This is going to cause increased mitochondrial permeability. So let's see how this happens. Now, due to the increase in cytoplasmic calcium, this calcium will now enter the mitochondria, as this calcium is taken up by mitochondrial calcium transporters such as the mitochondrial calcium uniporter. And as these transporters take up more and more calcium, which is very high in the cytoplasm now, more and more calcium enters the mitochondria, and now inside the mitochondria we have very high calcium concentration. Now this calcium concentration in the mitochondria, this high calcium concentration, is going to trigger the formation of this complex protein assembly called permeability transition pore, or PTP. This protein is going to assemble in the inner mitochondrial membrane. So we see these PTPs now assembling on the inner mitochondrial membrane. Now, because of the PTP formation in the inner mitochondrial membrane, protons as well as all the other mitochondrial chemicals less than 1,500 Daltons, they will start diffusing across the inner mitochondrial membrane, and that will lead to a loss of proton gradient, and without the proton gradient, oxidative phosphorylation is going to be affected, and this is going to cause further decrease in ATP. So this disruption of the oxidative phosphorylation, we now have an abnormal oxidative phosphorylation going on, and this is going to trigger the formation of reactive oxygen species. And we'll look at this reactive oxygen species and what it does to the cell in a future video. Besides triggering the formation of these pores in the inner mitochondrial membrane, calcium also activates several hydrolytic enzymes. So, for example, the protease called calpain is activated because of this increased cytoplasmic calcium. And calpain breaks down plasma membrane as well as other membrane proteins and also cytoskeletal proteins, these proteins that maintain the cell structure. Calcium also activates ATPase. This is the enzyme that breaks ATP down to ADP and PI, and that will lead to even further decrease in ATP. Calcium also activates the enzymes, the endonucleases enzymes, and these endonucleases will then work on DNA, break down DNA, and this will cause chromatin fragmentation. Another family of enzymes that are activated by calcium are the phospholipases. And as you remember from previous videos, phospholipids are important components of cell membranes. So phospholipases break down membrane phospholipids, and that leads to further membrane damage. So at this stage, under a light microscope, we see what is called a hydropic chains or a vacuolar degeneration pattern, where you see increased cell size. The cytoplasm has a pale eosinophilic stain. Eosin is a stain that stains cytoplasm. So there's a very pale staining rather than a dark staining. There are a lot of clear vacuoles in the cytoplasm. We can see that under a light microscope. And these vacuoles are basically pins of swollen parts of the ER, of the endoplasmic reticulum. If this was a liver cell, you will see these vacuoles filled up with a lot of lipids. So in the liver, this will be called a fatty liver. So that's a fatty chains. Under an electron microscope, we'll see an electron-lucent cytoplasm. That means the electrons easily pass through the cytoplasm because all the proteins in the cytoplasm are diluted since the cell is swollen. There's a lot of water in the cell. With the EM, we also can see chromatin clumping, as we mentioned earlier. We see the ribosomes becoming detached from the rough endoplasmic reticulum. That's where proteins are being synthesized. We see the ER becoming swollen with vacuoles that have become pinched off. Due to the water accumulation, we see dilated Golgi. Due to the protein breakdown, we see cytoskeletal damage, for example, loss of microvilli, and the formation of irregular bulges in the membranes that we call blebs. So here, for example, you see these irregular bulges in a membrane called blebs. We also see these damaged membranes that coil into whorl-like structures, what we call myelin figures. So here, for example, is a myelin figure. This is a myelin figure, basically damaged membrane coils into whorl-like structures. Another thing that we could see under the electron microscope at this stage would be amorphous phospholipid deposits in the mitochondria. So the mitochondria has these amorphous phospholipids that have deposited due to membrane as well as protein damage. There may still be some hope. If the blockage is removed at this point, the cell injury may still be reversible. For example, if we use thrombolytic therapy using drugs such as activates, which is a tissue plasminogen activator, as well drugs as streptokinase, these drugs will dissolve this clot. It will dissolve this clot, and that will open up the blood supply and bring in the nutrients and oxygen that this cell desperately needs. So this cell could, at this point, still go back to its healthy state, start making more energy because now oxygen as well as nutrients is available. And with all that energy available, the pumps will start working and try to reverse all the conditions that we just talked about. So that's why it's very important, because of this availability of thrombolytic therapy. It's very important that if somebody has a heart attack or stroke, to try to reach the hospital as fast as possible, because this thrombolytic therapy works very well if it started very early on, so that the cell injury is still reversible. If the person comes to hospital much later, then the cell injury becomes irreversible. The cell cannot restore the mitochondrial energy generation, and that's going to lead to severe destruction of the plasma membrane as well as other membranes, as well as breakdown of chromosomes. So once these three things happen, basically the cell cannot go back to its healthy state. And now the cell may undergo necrosis. So in necrosis, we see features in the cell such as increased eosinophilia, strong eosin binding. That's because of loss of RNA in the cell, as well as increased binding of this eosin stain to cytoplasmic proteins that have become denatured. So that's why we see a stronger red color of necrotic cells. We also see mitochondria that are swollen and may show precipitated calcium as well as protein deposits. Another feature of necrotic cells is the breakdown of nucleus. We see cariolysis, where there is faded nuclear staining due to DNA breakdown. We see pycnosis, which is nuclear shrinkage as DNA condenses. And then we may see karyohexis, where the nucleus is fragmented. So all of these things are going to cause release of these cellular contents out of the cell, and that's going to trigger inflammation. Instead of necrosis, the cell may also undergo apoptosis. Necrosis is due to pathologic reasons. Apoptosis, programmed cell death, may be pathological or physiological. In apoptosis, the morphology that we see is different from necrosis. Typically, in an apoptosis cell, what we see is the cell shrinks remarkably in size. The plasma membrane is intact. Okay, although the structure of the plasma membrane may change, the membrane is intact. Okay, which means the cellular contents are not being lost. And since the cellular contents are not being lost, there is no inflammation. Okay, apoptosis does not trigger inflammation. The nucleus is fragmented into nucleosome-sized fragments, and we'll talk about this in a future video on the mechanisms of apoptosis, and we'll see why this nucleus is broken down into pieces that resemble the size of nucleosomes. So once again, the cellular contents are intact. They may be released as membrane-bound apoptotic bodies. Okay, so these apoptotic bodies is how this cell will break down, but these are all membrane-bound. So important thing, because they're membrane-bound, nothing is lost outside. None of the cellular contents are lost outside. There's no inflammation. And the macrophages and other phagocytic cells present nearby, these can then eat up these apoptotic bodies. Okay, they can basically destroy these small apoptotic bodies, and that's how the cell can die by apoptosis. Now, one of the important ways we see we can detect cell injury in the body is through analysis of biochemical markers. Okay, so these are clinical lab tests. So for example, in myocardial infarction, when the heart cells are being injured, we can see increased levels of troponin, which is a protein found in the heart muscle. So troponins as well as creatine kinase MB are two tests that we do for heart attacks. If there was acute pancreatitis where the pancreatic cells are being broken down, then we can measure amylase and lipase in the blood, in the serum, and that will help us in the diagnosis of acute pancreatitis. Similarly, there are many other lab tests that can be used for various tissues and organs, and we'll talk about these lab tests in future videos. So hopefully this gives you an idea of cellular adaptation, injury, as well as death. In the next video, we'll talk about reactive oxygen species, free radicals, oxidative stress, which is one of the most common reasons why cells undergo injury. So this is a very important topic that we'll talk about in the next video. So until the next video, once again, thank you very much for supporting this channel. Hope we can help you understand the biochemistry as well as biochemistry of health and disease, and we'll hopefully see you soon in the next video. Have a wonderful day.